And next speaker is Dr. Gabriel Kreiman from Harvard Medical School, United States. And his title is Peeking Inside the Brain to Develop the Next Generation of AI. Please.
Thank you very much for the introduction, and I'd like to thank the organizers for a very exciting conference. I'm really enjoying learning from all of you, and also enjoying the beautiful city of Kyoto. So, domo arigato. So I'd like to tell you today about a problem that connects to many of the previous talks, and which has to do with being able to understand the types of brain computations that lead us to be able to perform visual recognition. So we take an image like this, and in a fraction of a second, we're able to capture a lot of what's going on in this image. In fact, in a couple of 100 milliseconds, maybe a few fixations, we can understand and answer a wide variety of questions about what's in there, where is Obama's food. We can answer questions like search for all the mirrors, what are they doing, where are they, and even sort of conjectures about what happened before, what's going to happen next, how many shoes are there, and so on. And so we can answer essentially an infinite number of questions on this image. And we'd like to understand, what are the brain mechanisms that allow us to perform this. We'd like to understand this at the behavioral level. And we're also interested in trying to educate computers and teach computers how to see. That is, building AI algorithms that can do the same things, that can pass a Turing test for vision. The idea is that one day we may be able to have a computer or a human will be able to answer any type of question, and we will not be able to distinguish whether that image was interpreted by a machine or a human. And I'd like to give you a glimpse of where we are in terms of trying to do scene understanding. I'd like to tell you a little bit about a lot of the exciting progress that has been happening in the AI domain in terms of these questions. And I also would like to emphasize how extremely far we are from achieving this type of goal, having a machine that can pass a Turing test for visual scene understanding. So to understand a complex scene, we think about a divide and conquer strategy, where we try to break down the task into simple, reusable visual routines. Visual routines is a term that was coined initially by Shimon Ullman, and refers to the idea that we may have simple elementary building blocks or simple computations that we can essentially plug and play into multiple places to solve different types of visual tasks. So this is a working hypothesis. This is a list of certain computations that we conjecture may be important for scene understanding. I do not think this as an exhaustive list. I do not think of this as a final list. I suspect that we'll have to revisit many of these different types of visual routines, and they may have to change in pretty strong and dramatic ways. But it's a useful way for us to think about what are the basic steps that we need to understand to do visual scene understanding. So these steps include things like extracting a sensory map, proposing a gist of the image, being able to fixate on a given object and propose what's the object at the level of the fovea, being able to combine all of the steps above to do inference, temporally store that type of information, task-dependent active sampling, essentially moving your eyes to a new location, new fixation. These steps can be repeated multiple times. And then eventually, in the context of answering a particular question, being able to decide, do I have the answer, yes or no, and produce that answer. So in each of these routines can in turn be subdivided into many smaller subroutines. So the one that we probably understand the most is foveal object recognition, the idea of having a particular image projected onto the fovea and having an algorithm like a deep convolutional neural network that will ultimately be able to provide a label to what objects present in the fovea. And this may amount to subroutines like querying different parts of ventral visual cortex. This is jargon for different places in visual cortex. Some of you may are very familiar with these regions, V1, V2, V4, integrating temporal context, integrating spatial context, and selecting a specific computation to do the classification, and so on. So what I'd like to do today is give you a flavor for some of these basic visual computations and visual routines. So I'll talk a little bit about some of our current understanding in proposing foveal objects and computational models of visual object recognition at the fovea. I'll talk a little bit about inference and how we can do things like pattern completion, and then also about task dependence. And if I have time, maybe I'll very briefly mention some of the work that we're doing by going back to the beginning and trying to look at contextual information and proposing an image gist of the image. So let me start by a problem that has bothered me for a very long time, and which I think we've made a little bit of recent progress recently, which is trying to understand what neurons along ventral visual cortex encode in terms of visual information. I refer to this question as what neurons in ventral visual cortex like. And this is joint work that I've done with a couple of amazing people, including monkey physiologists in Marge Livingstone lab, and a very talented grad student, Will Xiao, that did all the computational work. So a lot of what we understand about deep convolutional neural networks can be somehow mapped onto basic aspects of architecture and neurophysiology of visual cortex. So this is a picture that was flashed earlier by Professor Richmond before. This is a schematic diagram of the mesoscopic connectivity in the primate visual cortex. So this gave us the idea that there's some sort of hierarchy in processing visual information. And this was combined with about 5-6 decades of heroic experiments inserting electrodes in different parts of visual cortex to try to elucidate the tuning properties of neurons in different brain areas, starting with the seminal work of Stephen Kufler in the retina, discovering the receptive field properties of receptive fields of retinal ganglion cells, all the way to Hubel and Wiesel and orientation tuning in primary visual cortex, the discovery of tuning for motion, all the way to complex tuning properties, including a lot of work done here in terms of responses to complex objects like faces and so on. And this idea of hierarchy in terms of the architecture, in terms of the visual selectivity, was taken very, very seriously by computational scientists. In fact, you could argue that computer scientists took it even more seriously than neuroscientists. And that led to the creation of hierarchical computational models. Professor Richmond mentioned the early work of Professor Poggio, even before that Fukushima, Jan LeCun, and many others, developing computational models that respect some of the basic properties of physiological responses, as well as the architecture of visual cortex. And this provides a first-order approximation to the computations during the 150 milliseconds after stimulus onset in primary visual cortex. This, in turn, later on inspired the development of deeper and deeper convolutional neural networks. There's a wide family of architectures that share similar properties. Yuki Kamitani showed many of these types of architectures. And these are the kinds of architectures that are extremely successful these days in a wide variety of pattern recognition tasks in AI. These are the architectures that win all the competitions in computer vision in terms of visual recognition, in terms of labeling objects, in terms of clinical image understanding, and many other things. So I want to go back to the basic question of how do we know what are the tuning properties of neurons along ventral visual cortex. So I mentioned the classical work of many people in terms of trying to elucidate what kind of features, what kind of images trigger high firing rates in the brain. And one of the main challenges in designing and conducting these type of experiments is that we have to decide what kind of images to use to probe the responses of neurons. It is simply impossible by current or foreseeable technologies to be able to exhaustively sample stimulus space. So for example, if you have an image that's just 100 by 100 pixels, each one of those pixels is 256 shades of gray. This is, of course, a far cry from natural images. But even for this very reduced stimulus space, there are 256 to the 10,000 images. If you have a grad student that doesn't sleep, doesn't eat, and all he does is present pictures for 5 years, you would not be able to exhaustively sample stimulus space. He would actually have to work for about a millennia without sleeping or drinking to characterize the activity of a single neuron exhaustively in this way. So this is just not quite doable. So what we do is we have a number of heuristics and approximations, and I'm very excited about this recent work that was led by this talented student, Will Xiao, trying to come up with an unbiased method to investigate the tuning properties of neurons in visual cortex. So what he did was combine 3 different elements. One is a generative neural network. So he took one of these deep convolutional networks and he basically flipped it upside down. So in a typical deep convolutional neural network, you start with images and you extract features. What he's doing here is starting with features and creating images. So this is a way of generating images. Each of those images is presented to a monkey. He's recording the activity of a neuron in a particular part of the brain in monkeys, using the responses of that neuron as a fitness function to evaluate which pictures are good, which pictures are bad. By good and bad, what I mean is generating high spike counts in a predefined window after stimulus onset, and then recombining those features from those images using a genetic algorithm, and then using this in a closed loop fashion to generate to get another generation of images. So starting from noise, the idea is that the neuron itself can guide in a largely unbiased fashion what kind of images it likes or it doesn't like. So the idea is we will start from noise, we record firing rates, we genetically recombine those features, we create more pictures, we record firing rates, and we go on and on and on until we converge onto stimuli that trigger high firing rates. So let me show you an example neuron. This is an example neuron recording from macaque inferior temporal cortex. In fact, this is a neuron that was in the posterior inferior temporal cortex in one of the so-called face patches as determined by functional MRI. And what you're seeing here are the responses to about 2,500 different natural images. In the bottom, you can see the top 10 images. That is the 10 images that elicited the highest firing rate for this neuron, and the worst 10 images. And then on the right, what you see is the average response. So this neuron had a higher response to pictures of monkey faces and human faces compared to other animals or other objects. So based on this type of firing rate responses, some people would like to put a name to this type of neuron and say this is a face neuron. What I mean by that is that there's a somewhat higher firing rate to some pictures that contain faces versus other pictures that do not. So what happens if we use our synthetic image generation algorithm? So that's what Will and others did in here. So what you're seeing here is the response of this neuron on the y-axis as a function of the generation number. Generation is one loop of that closed loop algorithm that I mentioned. So we present a wide variety of natural images. Those are the 2,500 natural images that I mentioned before. Those include objects and faces and whatnot. And those are shown here in green. Basically, the responses to those images do not change much from one generation to another. Those pictures are fixed. The responses decrease a little bit over time, probably due to adaptation. Our algorithm can increase the firing, can generate images that will trigger higher and higher firing rates, as shown here in the black curve. And within about 50 to 100 generations, it seems to converge onto images that trigger high firing rates. So here are the early synthetic images from our algorithm. At the beginning, we start with noise, so we start with images that are very bad at triggering high firing rates. And at the end, we end up with pictures that trigger very high firing rates in this particular neuron. So what do those images look like? So let me give you an example. This is when we start with noise. This is the first generation of that particular run. This is the next generation, and so on. So after many generations, we create better and better images. Again, by better, I mean images that trigger higher and higher firing rates. So this is the picture at the end of when we stop this process in generation 209. And this is what it looks like. So some people may like to put names and try to argue about what those images actually represent. Here's a video just showing the whole process from the beginning, starting with noise up until convergence for another one of these of these neurons. And what I think we're seeing here is the idea that the kind of stimuli that can trigger high firing rates. So first of all, are different from the type of natural images that we and many others have been using to probe the responses of neurons along visual cortex for many, many years. And second, that maybe we do not need to map directly onto words, that describing the tuning properties of neurons using words in English or Japanese or whatever language you want is not the adequate language to describe what's happening. Ultimately, we need computational models. The language of science is mathematics. We don't want to use terms like this is a chair neuron, this is a face neuron, this is a curvature cell, because we may be able to have different types of images that trigger much higher activity, and those are ultimately combinations of features that do not necessarily map onto any single one human concept. So to summarize, we refer to this algorithm as XDream. This is a proof of principle algorithm that can generate visual stimuli. It's largely unbiased, or at least much less biased than anything that people have tried before. It covers a very vast stimulus space. It can work in real time. And importantly, we do not need, so just for the aficionados, we do not need to compute any gradients. We do not need to have any knowledge about the internal architecture, and that distinguishes us from many other similar approaches in the field, including what Google did in terms of deep dreaming and so on for people who know about this. So with this algorithm, we can generate what we call super stimuli for model units. So we can do this in computational models, and we can show that we can generate stimuli that are better than the millions of natural images available on the web. We can do this across different layers, different architectures. And we have also tested this with actual biological neurons throughout different parts of visual cortex, most prominently in V1, posterior and anterior parts of inferior temporal cortex. And we can generate images that trigger at the firing rates that are at the same level or even better than the kinds of natural images that we and many other people have been using. These are largely invariant. The responses to these images are largely invariant to changes in scale and location. And this can reasonably well approximate the similarity to these images can help us better understand the responses to natural stimuli as well. So this is our initial attempt to think about what neurons like in computational models of foveal recognition in cortex. And I'd like to switch gears now to what we think of as the next step in this type of computations. I seem to simplify the problem. Sometimes I distinguish between what happens in the first 100, 150 milliseconds of processing and what happens in the next 50 to 100 milliseconds of computation after the initial glimpse of an image. And we refer to this problem as inference, meaning that we can take now all of these visual inputs and then combine them in some sort of form with prior knowledge to do visual recognition. One of the problems in which this comes about continuously during natural recognition is to be able to interpret objects that are heavily occluded. During natural vision, a large fraction of the objects that we see tend to be either partially or very heavily occluded, as you can see in this image. And you can still recognize these chairs and these tables in the Piazza San Marco in Venice, despite the fact that you may have never been here before, you may never have seen this kind of place that's completely flooded, and yet even with this very simple exposure of a couple of pixels, you can still make inferences about what those objects are. So we did a study to try to evaluate the process of pattern completion, the idea of being able to make inferences about objects that are from minimal pixel information. So let me start by showing you some behavioral data describing how robust visual recognition is to heavy occlusion. We used a technique called bubbles, where you can see here on the left a toy school bus. When you have many bubbles in the images, it's pretty easy to recognize. You have lots of pixels, lots of information. When you have very few bubbles, it becomes harder and harder to recognize the object. And that's what you're seeing here. This is behavioral performance in a 5-way forced choice categorization task as a function of the percentage of object visibility. When the objects are 100% visible, this is a very simple task. People are essentially at ceiling. When the objects are, when you have only 1% visibility, subjects are essentially at chance. The colors here don't matter too much. This is the amount of exposure time for the objects. So you see that humans are extremely robust to heavy occlusion. They can still recognize objects well above chance. Chance is 20% in this case, even when you have up to 20% or 10% visibility. We conjecture that, based on previous work, that being able to do this type of inference and pattern completion requires additional computation time. And we used a very simple behavioral trick to try to test this idea. And this refers to using backward masking. Backward masking refers to the idea of injecting a noise mask very briefly after presenting the stimulus, and is purported to interrupt visual processing. The idea is that you have flow of information from the object, and the mask, the noise, interrupts those computations. So this is the behavioral data that I showed you before, and this is what happens when you quickly introduce a visual mask. The color of the curve now does matter. So the color of each curve now indicates for how long the stimulus was presented before the introduction of the mask. So if the stimulus was presented for about 150 milliseconds, that seems to provide enough computational time for recognition, and nothing much happens in terms of backward masking. So presenting the backward mask at 150 milliseconds is just too late. But if you present the backward mask at 25 milliseconds after stimulus onset, then people are essentially at chance. So people are not able to do pattern completion, to be able to infer what the objects are when they are heavily occluded, if they have minimal computational time as interrupted by backward masking. So this was the first cue for us to think about that there may be additional computations that are needed. There's an additional cost, there's an additional time that's needed to be able to do pattern completion. Let me show you quickly what happens at the physiological level. I'm going to switch species now in terms of physiology and show you a little bit of human neurophysiological data. These are data that come from patients that have epilepsy, and they have electrodes implanted in their brains in order to localize where the seizures are coming from. So these are recordings. In this particular case, these are intracranial field potential recordings. These are not single neuron recordings. These are field potential recordings inside the brain for an electrode in inferior temporal gyrus, presumably a homologous region to the type of macaque inferior temporal cortex recordings that I referred to earlier. What you see here is the intracranial field potential as a function of time when presenting a fully visible image. This is typical for many of these recordings. We and many others have shown this before. We have very rapid, about 150 millisecond response, highly selective responses, highly reproducible responses in response to these type of pictures. So when we present heavily occluded images like the ones that I'm showing here, we still see a similar response. The responses are not quite identical to the fully visible counterparts, but despite the fact that there is very heavy occlusion here, despite the fact that the subjects are only seeing about 10 to 20% of the pixels in the image, we can still recover a selective response from this electrode, even in single trials as I'm showing here. The numbers that you see below these figures indicate the latency of the response. The latency in the response to the full image was 150 milliseconds, in this case defined by the time of the first trough in the voltage signal. And the latency in the response to all of these other trials was anywhere from, let's say, 190 all the way to 290 milliseconds. In other words, at the physiological level, there is also a cost. There's also an additional time required to do visual recognition and pattern completion. I just showed you a handful of trials. Let me show you now all the trials for this one electrode. These are all the responses in single trials for the fully visible whole objects. These are all the responses in single trials for the same electrode to the partial images. So first you see that there's more variability. These images change from one trial to another. But most importantly, you see that there's a significant delay in the responses. It costs about 50 milliseconds to be able to do pattern completion, both at the behavioral level as well as at the physiological level. So next we tried some of the state-of-the-art AI algorithm for computer vision. In black you see here the data that I showed you earlier, the human behavioral data, and then all of these colors represent to a wide variety of deep pool convolutional neural networks that are essentially state-of-the-art and can do extremely well in a lot of visual object recognition tasks. So you see that none of these computational models, none of these purely feed-forward state-of-the-art computer vision models can reach human-level performance in terms of pattern recognition. So inspired by the behavioral data and the physiological data, we conjecture that this additional time that's needed for pattern completion essentially amounts to using recurrent connections. The idea that you need additional computations that may be instantiated by either horizontal and or top-down connections that are ubiquitous throughout visual cortex. So what we did here was we took one of these deep convolutional networks and we took the top level of that model. In this particular case, this is the AlexNet model that Yuki was also referring to before. And we took the top level of that model, the so-called fully collected layer 7, and we injected all-to-all reciprocal connectivity at the level of FC7. We've done this in many other models, and this is just one illustrator. So what this does is create an attractor-based recurrent neural network in just the top layer of these computational models. And that's the model that I'm showing here as RNN-H. H refers to Hopfield model. This is a well-known attractor-based recurrent neural network. And we see that just by injecting these recurrent computations at the top level of the hierarchy, we can already improve performance from the computer vision systems to approximate human-level performance. If we further allow ourselves to train the weights in those recurrent connections, that's the model that I'm showing here as RNN5, where we allow ourselves to change the weights and train them, then we can achieve essentially human-level performance in this pattern recognition task. So to summarize this part and to simplify the discussion, we think that the visual system may operate in two modes. One is which is essentially feed-forward and extremely fast. This may account for the types of computations that you're able to conduct if you only have 100 and 150 milliseconds of computation. And then there may be slower modes that require horizontal and or top-down recurrent computations that may give us a first glimpse at what happens during the next 50 to 100 milliseconds of computational processing before you make the next saccade. So to summarize, visual recognition is robust to heavy occlusion. Pattern completion requires extended computations. We think that this costs on the order of 50 milliseconds, both in terms of behavior as well as in terms of physiology. Current state-of-the-art feed-forward networks do not seem to be able to account for this type of inferences during pattern completion, and we think that we can explain the process of pattern completion, at least to a first approximation, using recurrent connectivity and horizontal connections. This is just a disclaimer. I am not claiming we understand every aspect of pattern completion. There is much more to pattern completion than what I'm describing here, and I'm happy to take questions and discuss this in more depth. Okay, I want to very quickly tell you about one more step in this hierarchy of routines that we think is needed for visual recognition. So we have very fast feed-forward processing that costs about 150 milliseconds of time. Then we have additional recurrent connections that may give us the next 50 to 100 milliseconds or so. And then the next step is to move your eyes. You're examining a visual scene. You have to make a saccade to a new location. And we'd like to incorporate the ability to move your eyes to a new location into our computational models. So for that purpose, we introduced a simple visual search task. So first of all, just let me show you, this is something that probably many of you are familiar with. When you're examining a scene, you're constantly moving your eyes. Depending on exactly what you're doing, you're moving your eyes about 3-4 times a second. These are actual recordings of eye movements for this particular image from one subject over 12 seconds. So to study the problem of eye movements and to try to build the computational models that can predict eye movements in natural scenes, we used the visual search task. So this is one example of a classical visual search problem. This is the where is Waldo problem. Waldo has different names, I learned, in different cultures. I hope some of you can tell me what he's called in Japanese. So whenever I present this picture, somebody wants to know where Waldo is actually in this picture. He's actually right there. We want to be able to have a computational model that can predict visual eye movements and visual behavior during visual search, and that means explaining at least 4 key properties of visual search. One is selectivity, to be able to distinguish the target, in this case Waldo, from all the distractors. Invariance, we want to be able to find the target irrespective of changes in appearance. If Waldo is larger or smaller, or if you would rotate Waldo, we should still be able to find it. We want to do this efficiently without having to scan the entire image pixel by pixel. And importantly, we want to be able to do it with, in a way that we can generalize to novel situations. We want to do it in a way that has essentially zero-shot training without having to train the system for every new object that you have to search. So we conducted 3 different visual search experiments. The first experiment is with object arrays. There are 6 objects on the screen. We present the target, and subjects have to move their eyes to find the target object. In the second experiment, we did the same thing but with natural scenes. There's a target object, in this case Minnie Mouse, and subjects have to move their eyes to find Minnie Mouse in that complex scene. And finally, just to torture subjects, we use a real, actual Where's Waldo experiment. Subjects hate to do this. It takes forever to do it, but we scan people's eye movements during this task. So here's behavior in the first experiment, the one with object arrays. What I'm showing you is the cumulative performance as a function of fixation number. So because there are 6 objects, if you're randomly moving your eyes, this is what chance levels are. This is how fast you should be able to find the object. This is actual behavioral data from 15 subjects that participated in this experiment. So people are well above chance in performing this task. As a minor comment, after 6 fixations, people are not exactly at 1, and that is because humans are funny and stubborn creatures. Sometimes they move their eyes back to the same location, despite the fact that they know that that's not where the target is, but they do that anyway. So let me give you a quick glimpse of a zero-shot computational model that we propose to describe that next step of computation, meaning how people move their eyes in a task-dependent manner. So we start with a picture here, the picture of the target that we're going to look for, and that image goes through a deep convolutional neural network that was pre-trained for visual object recognition. So we're not training anything based on our images. This is a convolutional neural network that was trained for another task, for object labeling. In this particular case, we're using VGG16. Again, Yuki introduced this network before. We need to be able to remember the features that we're looking for. So we introduce a module that we refer to as prefrontal cortex. This is just a loose term that we use to define working memory, the ability to be able to remember that target in order to look for it. Next, we have a search image. That is where we're going to be looking for the target object. That image is passed through the same exact network without any retraining. It's exactly the same network with exactly the same weights. We extract features. And then we have top-down modulation. You can think of this as top-down feature-dependent attention that essentially convolves the target the features of the target that we're looking for with the features throughout this network. That creates an attentional map, and we use a winner-take-all mechanism to select the location of the next fixation. So thus, the model can select one particular place in the image to move its eyes. And then we have, in the simplest version, we have infinite inhibition of return. That is, the model remembers exactly where the target was, where the fixation was. If the target was there, search stops. If the target was not there, then we select the next maximum, and so on. So in that way, we introduce a very simple form of dynamics into the model. There's no training in the model. There are no free parameters in here. The model can get any target image, any search image, and produce a sequence of eye movements, and we can compare that against human levels. So this is the data that I showed you before in terms of cumulative performance, a function of fixation number, and this is what our model does. The very close similarity between the model and humans, I think, is largely coincidental. I'll show you in a second that for the other experiments, it doesn't look as nice as this. What I do want to emphasize is that the model can perform visual search with these images, despite the fact that it was never trained to do visual search at all. There's a large number of alternative null models that we've used for comparison. This is not just pure bottom-up saliency. This is not just pure template matching. We can discuss some of these controls later. This is the same results for the experiment 2. This is chance levels. This is here with natural images. This is human level performance. This is what the model does. As you can see, we're not trying to match human performance. We're not fitting any parameters in here. Here, the fit between the model and human is certainly much worse than before, but still the model is able to capture and perform visual search in this complex task, despite the fact that it was never trained to do this either. Here's the same plot for the actual Where's Waldo images. And again, the model is able to search for Waldo, despite the fact that it has never seen Waldo before. That is, there's no training with Waldo. These are all cumulative performance values, that is average number of saccades required to find the target. We've also done trial-by-trial comparisons of the exact spatiotemporal sequences, that is exactly at which position, at which time people move their eyes, and the model moves its attentional target, and I'm going to skip that in the interest of time. So just to summarize, humans show the 4 key properties of visual search. They have selectivity, they have invariance, they are efficient, they can do this without having to scan the image pixel by pixel, and they can also generalize to completely novel objects that they haven't seen before. I didn't show the psychophysics data for that. We proposed a very simple invariant visual search network that has 0 free parameters. It's inspired by basic aspects of neurobiological architecture, and it can perform target-dependent, feature-based top-down modulation to do visual search. And again, there's much more to visual search. There's contextual information. There's temporal information, temporal integration, and memory. And these are many things that we still have to incorporate into this type of computational models. I'm going to skip the last part. I just want to give you a quick glimpse of what I mean by proposing the image gist and how contextual information may be important for visual object recognition. I'm just going to do that with a quick example. So if we're trying to recognize what this object is, I don't have time to do a quick psychophysics experiment here, but trust me, this is a pretty hard problem. Most people don't know what this object is. However, if you are presented with the whole image, even if you don't move your eyes, even if you are fixating on the object, then it becomes pretty easy to recognize. So this is what we mean by the gist of the image. Some basic intuition about what's in the context, about what the rest of the image looks like, can be extremely important for visual recognition. And we're trying to model how it is that humans are able to incorporate this type of information. This is just another example. If we flash an image like that, it's very hard to recognize. If you flash the entire image, it becomes much easier to recognize that object. So we think that contextual information is very important for everyday visual object recognition, and we're trying to model that. So I'm going to skip this just to quickly summarize the work that we've been doing in context-dependent visual recognition that I didn't get to show you any of the results. We think that contextual information plays an important role in visual recognition. Context has to be at the right time, at the right place, in the right amount. And we can even guess what an object is without any information about the objects, purely based on contextual information. We also have a computational model that tries to incorporate contextual information into visual recognition. If anybody's interested, please come and talk to me about this. So I want to quickly close by giving you a quick flavor of where we are in terms of AI and scene understanding. And I'll do that by showing one example of a task that's very common these days in the computer vision literature, and it has to do with image captioning. You can actually try it yourself. There are many systems out there for image captioning. This one in particular is from Microsoft. This is the Microsoft captioning bot. You can take any picture that you want, you can upload it on the web, and the system will try to describe it with a short sentence. So if you put this picture onto that system, I uploaded this picture, and the system says, I think it's a group of people standing in front of the Leaning Tower of Pisa. So this, I thought it was very impressive. This is well beyond where computer vision was about a decade ago. So the system can recognize the Tower of Pisa. It recognizes that there are people. It recognizes that those people are standing. So this is quite amazing. Now, if you think about it, there are probably gazillions of training examples of the Leaning Tower of Pisa. Tourists are not super original in the way they take those pictures. There are probably many pictures that have the Tower of Pisa in exactly the same angle, with exactly the same cloudy conditions in the sky, and so on and so forth. So there may be a lot of training data to be able to do this sort of thing. But regardless, this is quite impressive. There's a lot of object labeling that these systems can do pretty well. But look at this picture. So I uploaded this picture now, and the caption for this picture was, I think it's a person standing in front of the Leaning Tower of Pisa. So again, it's pretty amazing. It recognizes the Tower of Pisa. It recognizes there's a person, a single person in this case, and so on. But I would say that the system missed completely what's actually happening in this scene. So this picture is meant to be semi-humorous. There's a perfect alignment of the ice cream cone with the Tower of Pisa. It's making you think that the Tower of Pisa is the ice cream. She's sticking her tongue and licking the ice cream. So all of these fundamental aspects of what's happening here in the scene are completely missed by the system. I think we're nowhere close to be able to do scene understanding to a level that a computer vision system can take this picture and understand what's going on here. If you scramble the picture, the system says, I think it's a picture of a person standing in front of a building. So again, here the system misses completely what's going on just by doing a very simple scrambling of the image. So let me take many steps back and conclude with a quick overview of where we are in terms of vision and scene understanding. So there has been enormous progress in computer vision and computational neuroscience of vision. So there are certain problems that, if you think about it purely in terms of labeling objects in the image or succeeding in terms of performance, I think these problems are essentially solved. We have systems that are better than humans in terms of reading barcodes, face recognition. We have computer systems that are better than humans in face recognition. Classifying images like birds, galaxies, tumors. These are all problems where computers are already better than humans. This does not mean that we understand what's happening inside the brain at the neuronal level, but in terms of just performance, I think these are problems that are essentially solved. And yet, I would argue that machine intelligence is still very far from biological intelligence. They're extremely susceptible to different types of tricks and noise. Some of you may be familiar with adversarial examples. I think in terms of scene understanding, we have a very long way to go. And one example that I always like to do to mention is based based on the Obama picture, and the Tower of Pisa is understanding humor. The ability of taking a picture and understanding whether that picture is humorous or not. I think we're extremely far from being able to do that sort of thing. So I gave you a few examples. I think the path forward, and this is not a consensus in the field, many people would disagree. I think the path forward is to better understand neural circuits. I think if we can better understand how our brains solve these problems, we will be able to inspire the next generation of AI circuits. Why do I think that? Well, because our brains are the product of millions of years of evolution. They are efficiency in terms of the cost, in terms of power, in terms of size, and they're extremely robust. Robustness is probably a critical function for the evolution of our brains. So I think the robustness of our brains implies that we will not be susceptible to the same kinds of tricks and noise that current machines are. And therefore, I'm extremely excited about the future of what we might achieve at the interface of neuroscience and AI. And I'm going to stop there.
Thank you very much for your very wonderful talk. Yes.
I wanted to come back to the beginning of your talk on the XDream method. And what I'm wondering is, after you've characterized and found these large responding images, can you invert what you've learned so that if I handed you an arbitrary image, you could predict the firing rate of the neuron? Could you use it as a response model?
Yeah, that's a great question. The answer is somewhat. So here's one thing that we've done. So you can give me an arbitrary image, any picture in the world, the Tower of Pisa, the Obama picture, and I can compute the similarity between that picture and our preferred image. Now you can ask, well, what do you mean by similarity? So we can talk about what I mean by similarity. One version is the Euclidean distance in pixel space. That's not a very good metric of similarity. Another one would be perceptual similarity. Another one would be similarity in the FC7. But we have some measure of similarity, and that's a very simple semi-predictive model of firing rate. So we've done that not with the Obama picture, but we've done that with other pictures. We compute the similarity to the synthetic images, and that provides a decent explanatory power of firing rates. What do I mean by decent? Well above chance, but definitely we're not explaining 100% of the variance. So we can explain something like 40–50% of the variance in the responses of firing of inferior temporal cortex with this very simple similarity-based model. What we're very interested in doing now is going back and taking these images and going back to building better and better models. In this case, building better computational neuroscience models of visual recognition based on the responses to this type of evolved images.
Okay, I'm sure you may have more questions, but the time is limited. So thank you very much.